Good evening. Welcome back to Winchester Academy's Winter-Spring 2021 series. My name is Ann Berger-Linden, and I'm the Executive Director of Winchester Academy. We'd like to take a moment to thank the City of Waupaca and our producer, Josh Werner, for providing the space and technology to bring this program to you. Our next program, which will be the last of our Winter-Spring series, will be in 1 week on Monday, April 19th, same time, 6:30 p.m. Hannah Bukevich will be giving us a talk on the summer diet of wolves in Wisconsin. It's kind of a related program to tonight's. Tonight's program sponsor is Dr. Terry Hankey. Thank you, Terry. We will have a Q&A session following Lettie's presentation tonight. Questions may be submitted via the Facebook Live site or by telephone. The telephone number is 715-942-9917. I'll repeat that for you, and we will also announce it at the end of the program. 715-942-9917. Now it gives me great pleasure to introduce tonight's speaker, Lettie Vierk. Lettie is a native of Waupaca and graduated from Waupaca High School. She's currently a junior biology student at UW Stevens Point and intends to pursue a career in public health, specifically in epidemiology, once she gets her master's degree after graduating. Please welcome Lettie Vierk.
Here on my first slide, I have pictured Buddha for religion, a hospital for health, and the Democrat and Republican Party for politics. Now, what do these three pictures all have in common? Well, they are all topics that are pretty much forbidden when you first meet a stranger. And today we are going to be discussing a fourth forbidden topic, and that topic is going to be poop. I dedicated my summer of 2020 to analyzing the prevalent parasites in the Wisconsin wolf population. My name is Lettie Vierk. I am a junior biology major at UWSP. And here on my first slide, I have pictured Leishmaniasis. Leishmaniasis is my favorite parasite. It is transmitted by the female sandfly, and luckily it is not here in central Wisconsin. However, it still has the capabilities to affect canines. My research took place at the Waupaca Biological Field Station, and support for my research came from the Deborah Ann Martin Memorial Fund Grant Program. So you may be asking, why wolves? Well, there's already prior research going on at the Waupaca Biological Field Station, so I was able to kind of ask my own questions, branching off of the research already being conducted. And there also has been a recent increase in Wisconsin's wolf population. Also lucky for me, parasites are everywhere. They are in almost every single organism, and specifically for wolves, they like the oral anal route, and that is actually the most easiest for identification and classification, just simply due to the fact that these are going to be your larger parasites. And oral anal route, as you can imagine, is through the mouth and out through the other end. So what does life really look like for a parasite? It is a continuous cycle. They go through growth, reproduction, and transmission. This cycle is continuous, and the growth and reproduction is just like when a human being is going through puberty. And it's really important to understand that the transmission factor of a parasite life cycle is only a short amount. So the parasite isn't necessarily going to be contagious the whole entire life cycle. So what exactly is a host? Well, a host is an organism that allows for a parasite to not only survive, but also reproduce using the nutrients provided from that organism itself. And for parasites, when it comes to life cycles, we classify them in two different ways. We have direct versus indirect. Direct parasites are going to go from point A to point B, and that would be between two different organisms, and they won't try to infect any other host in between. Otherwise, most likely their life cycle will not continue. Versus our indirect parasites, which will use many different hosts throughout their lifetime to get from point A to point B. And that's what we would consider a wolf, as well as a human being. They would be considered an intermediate host. And intermediate hosts are on that roadmap of the parasite life cycle to their definitive host. So going into my research, I had two big questions that I would like to answer. And that was, what are the prevalent parasites in the Wisconsin population? And what does this tell us about the environment here in Wisconsin? For my research, I started out with the collection of fecal specimens, and this step was very important due to the fact that there's a lot of natural decay happening with the samples that I collected. I needed to make sure that I got to the centralmost part of the sample that was provided for me to make sure that the degradation of the parasites themselves weren't already too far for identification. And I collected about a walnut size of scat, and I did a 1-to-3 ratio of preservative, and that ensured a thorough preservation of my sample. I took gross examinations of all of the collected samples. This is important just to get as much information as we can possibly get, since this is not studied often at all. And the preservation of my parasites, I actually have a few different techniques for that. And also for the preparation of my slides, I had to do a few different techniques simply due to the fact that parasites are so different and there's such a wide variety of them. To visualize all of the parasites I was looking for wasn't going to be accomplishable by just using one procedure. Once I was able to actually preserve my specimens and prepare them for the slides, I was able to transfer the specimens onto my microscopic slides, dye them, and then I could actually start the identification process of the parasites. Here I have pictured one of my collection tubes. It's a 45 milliliter tube, and these are all reusable. So it may seem like a lot, but it's actually very simple. So we start with my sample collection. And for my sample collection, I worked hand in hand with Hannah Bukevich. She is studying the summer diets of the Wisconsin wolf population, and her research was done driving over 6 hours every single weekend during the summer. And she would find scat on the side of the road and actually measure it, the diameter of it, to make sure it was wolf scat that we were gathering. And we were mainly tracking 4 packs in the Central Forest, Wisconsin area. And the collection methods were really opportunistic roadway samples. She was mainly just driving around. There wasn't a lot of any other activities involved. In my preservations that I used, I started with a polyvinyl alcohol, also known as PVA. This is very highly regarded as a preservative. However, it doesn't necessarily preserve as long as I would like it to. The 10% formalin mixture I switched to about 25% through my research process, ensured that my parasites would be preserved for longer than I'll be alive, and our 10% formalin mixture is used in pretty much every single lab across America for preservation now. After preservation, we moved to the slide preparation, and I had to use different techniques for my slide preparation due to the different parasites. I had to use the direct smear versus the flotation technique. I have pictured on the right side of the screen, you'll see bigger particles underneath that cover slide, and that is from my direct smear sample. That ensures that I get the heavier eggs and the larger parasites that are more likely to float in my preservatives. And I also used the flotation technique, which was a hypertonic solution, as well as centrifugation, in order to float the eggs and the smaller parasites up to the top. I was able to use an inoculation loop then to gather the parasites off the top of my tube and actually transfer them to the slide. And you can see that on the left side, that fluid contains, compared to the particles, a lot more eggs compared to actual organisms. For the identification of the macroscopic organisms I found, this is actually a segment of a tapeworm, and I had to dye it and use the squish technique to actually identify the specific parasite that it was. And the squish technique allows you to actually squish the eggs out of the sacs, and then you can look at the eggs and identify the parasite through that. And so you actually take the slide itself and you put the cover slip on top and you squish it together, not enough to break the glass, but enough to just rupture those egg sacs inside so you have thorough viewing of the eggs themselves. And it's very important to understand that when I'm talking about the intestinal helminths, there's 3 big different classifications of these. We have nematodes, which are going to be your roundworms. They are non-segmented. We also have trematodes, which are going to be our flatworms and liver flukes. And we also have cestodes, which are going to be our tapeworms, which are segmented worms. So my main discovered parasite grouping are nematodes, trematodes, cestodes, and protozoans. Here I have pictured some nematodes that I found through my research. You can see a very pretty roundworm right there, and you can actually see to the left, on the left picture, on the left far side of that picture, an egg sac. You can see multiple black dots, and that is an egg sac of that exact roundworm. And I also have pointed out more eggs that actually contain larvae, and you can see those larvae inside. And on the right of the screen, I have a nematode in a cyst, and you can see it's non-segmented, and it's got a very nice, smooth, round body to it. Here I have some more pictures. The nematode shown on the left, most likely, when it was in the hypertonic solution, shriveled up a little bit, so it's a little bit harder to identify, but that is actually a worm inside of the egg there. And I also have pictured a nematode head, and you can actually see the little hooks on the very, very tip of it, and that's what the worm uses to actually latch onto you when it is inside of you. So this is actually a parasite I found specifically in my wolf sample, and this is the threadworm that typically infects most canines and human beings. I found one report actually recording this threadworm found in wolves, but that study was out of Europe. This is, as far as I know, the only one necessarily found in the US so far. I also have Toxocara canis. This is going to be the typical roundworm infection you're going to find in many canines, especially domesticated canines. It's very prevalent out in the wild, and you can see the larva actually inside of that egg there. And the shape of the egg is showing me that this is fertilized. And it's really interesting that this is fertilized because that proves to me that this is a current infection inside of the host. If the egg was not fertilized, that means that there would be external maturation happening, and so it wouldn't necessarily be a live infection inside of the host. Next, I will show you a few trematodes. Trematodes, typically you will not find the full bodies with my techniques I used. However, you can see quite a few eggs that I found, and you can tell that there's vast differences between these 2 different eggs. Just by looking at the center, that's the main identification marker for trematodes, is the outer layer and then the inner layer. On the left side of the screen, you can see that there are smaller circles inside of that egg, And on the right side, you can see it is just a singular unit inside of that, and the shape is very much different between the two. Here I have pictured another trematode egg, and you can see it almost looks like a perfect egg yolk inside that will be developing, well, it would have developed into the actual adult parasite itself. Next I have cestodes, and this was actually a cestode I found in the specimen that was provided to me, and that is a tapeworm. That segment that I found was about 3 inches long, and that's super exciting because typically with cestodes, they're actually made to be able to survive being pulled apart. They have their own reproductive systems as well as their own musculature systems inside of each segment. So that allows for any of those segments to be able to reproduce as well as move on its own. So if a worm gets broken off halfway through the defecation process, well then that other half of the worm will continue to survive inside of the host, and the other half that is left in nature will most likely be picked up by a bird because it'll be able to wiggle around. I actually was able to collect the exact sample from this specimen and find the cestode egg that is responsible for this worm, and that is pictured on the right side of the screen. It is kind of difficult to see, but there's a little circle inside, and that's actually a little larva all wrapped up in there. And one of the other studies I found, it was out of the US. That was the only study I was able to find out of the US. It came from Alaska, associating parasites and wolves. They found the hyatid tapeworm very abundant in the wolf population, and this was very intriguing to them, and I actually found it also very abundant in the wolf population that I had. You can see here I have cysts as well as eggs. The eggs are going to be the smaller circles on the screen, and as you can see, those are a little bit more clustered because they are used to being in an egg sac. Then I have the cysts also pictured. Those have the arrows pointing to them, and inside of those cysts will be containing larvae. I also found Cryptosporidium, and I'm sure most of you have heard of Cryptosporidium because not too recently there was an outbreak in Milwaukee. The interesting thing with Cryptosporidium, and I'll get into this a little bit later in my presentation, but it almost looks exactly like a pine pollen underneath the microscope. So just to make it clear, we have for intestinal parasites our helminths and then protozoans. They're two different species of parasites, but underneath our helminths we have our nematodes, trematodes, and cestodes. Again, our nematodes are roundworms. Those are non-segmented. We have our trematodes, which are our liver flukes and our flatworms, and we also have our cestodes, which are going to be our tapeworms, which are segmented worms. So to understand the parasite a little bit better, it is very important to understand the life cycle of the parasite itself. For the life cycle of a nematode, it actually starts off, the adult worm is living inside of the small intestine, and that is the growth factor of the parasite specifically. Once it actually gets out into the nature through the feces, that is when the external maturation is going to occur within the egg, and that is a giant step for growth within the parasites. And once there is the external maturation, it will then have to be ingested by a first intermediate host. And the first intermediate host can be any animal that provides a okay breeding ground for this parasite to survive. And you may be thinking that stomach acid, specifically with the ingestion of parasites, would be harmful, but stomach acid actually is advantageous to parasites because it allows for the egg to release the larva inside of the body. So the larvae essentially become free-floating inside of the body. And that's important to understand because the larvae will then use our circulatory system to work their way throughout our body to find a home to live in. Once they start to find a spot within our body that provides the nutrients they need, they can then move to reproduction. And then they will continue through that cycle to transmission, and that is just a continuous cycle. Here I have it pictured so you can actually see it. You can see starting off at number 1, that is going to be our definitive host, and that's where the adult worm is going to be living. And on the bottom at number 2, you can see the external maturation part. That will be our growth factor for the parasite. Once it makes its way to the first intermediate host, you can see it is the rabbit or the duck. That is when the reproduction will really start to take up with the parasite, and then the transmission occurs in their life cycle to get back to the definitive host. And you can see it's just a continuous cycle for these little guys. The life cycle of trematodes is very different. Trematodes are typically found in the liver, and that is just simply because for their organismal needs, that is the best organ that provides the nutrients for them. So the adult trematode will be living inside of the liver, reproducing, and the natural way to get out of the liver in most systems is through the gallbladder. So the eggs will be released through the gallbladder, and once the eggs are released through the gallbladder, they'll be introduced to the small intestine and then introduced into nature. And the trematodes, it's a little bit more specific for their life cycle. Their first intermediate host needs to be a snail. And if they don't make it to a snail, they will not survive. Once they finally make their way into the snail, and the snail could be infected through water, ingestion, fecal transmission, once it actually makes its way into that snail, that's when the reproduction will start occurring with this parasite. And it's going to form many different babies inside of this snail. So eventually this snail becomes just pretty much filled with this parasite. So hopefully a fish will come along and eat the snail, or the parasite will be released into the water. And once it makes its way to the fish from the snail, that's when the musculature of the fish will actually start to be infected. And so the snail is our first intermediate host, and then we work our way over to a fish. And then once it is inside of the fish, that is when it'll make its final step to the definitive host, and it will actually become transmissible to the definitive host. And it's very important for this trematode to have these specific organisms in its life cycle, because it will not be able to survive or reproduce if it does not have these intermediate hosts. It's also very important that the trematode ends up inside of the liver and gallbladder, otherwise it will also not be able to survive. Here you can see it. It is a smaller life cycle than I showed you before, and there is a water component to this parasite. The water allows for the actual organisms that are needed to be present. And what you'll notice for parasites, a lot of their life cycles are very circumstantial. They need to be at the right place at the right time in order for survival to continue for them. And for cestodes, the life cycle is more similar to a nematode. However, they are far more advantageous when it comes to being able to utilize their environment. And just like I told you before, their segments are actually made to be individually or survive individually. And that is far more advantageous than most parasites we see today. So the adult cestode will be residing in the small intestine. And once it is in the small intestine, that's where reproduction occurs in the definitive host. And these eggs will then be released into the environment. And I'm sure you can see a common theme of eggs released through feces, and that's why I was fortunate enough to get my fecal specimens. So the eggs are released inside of the feces also. And once the eggs are actually released into the feces, there needs to be more of an immediate host that comes to pick up these eggs. And that's also important to touch on because in the natural elements, and that's what's made part of my research a little bit more difficult, is that there's a lot of elements that are going against me as far as maggots, there's flies, there's rain, a lot of things. So the eggs are released in the feces, and then there is an ingestion of these eggs. And that could be from the actual cestode segment itself or just simply the eggs in the feces. And once that host is infected with those eggs, that's going to be our first intermediate host. Our first intermediate host will then begin to have those eggs start hatching. And this is similar to the free-floating analogy I used for nematodes. The stomach acid will allow for the eggs to actually hatch inside of the organism. And this allows for the penetration of the intestinal wall. And that's one reason why these parasites are a little bit more advantageous. They're actually able to, burrow through our body a lot easier than other animals. And that allows them for a more direct transportation to our bloodstream and being able to really transport everywhere in our body. And we see by far more detrimental effects of cestodes simply due to the fact where they can pretty much reach anywhere in our body. They can accidentally get to our brains and our eyeballs, but that's not where they need to be, so they'll eventually die. But once they make it into the small intestine and they're able to exit and enter as they want, there's actually going to be cysts that are starting to form throughout the body. And typically this is going to be in the musculature also, simply due to the fact that's what a lot of other organisms are going to be ingesting on the host. And once this host is pretty much just a big egg sac of the cestodes eggs, then the definitive host is going to ingest that intermediate host. And that could be a rabbit, it could be a pig for human beings, it could be a cow for human beings. Here you can actually picture the life cycle of the cestode. It is a little bit more simple. However, we do have intermediate hosts with it. You can see at number 1, we have the adult worm, and that would be residing in the small intestine. And that is when the growth and reproduction will occur. And at number 5, you can actually see that's where the transmission starts to occur with this parasite. So for protozoans, they have a far more different life cycle than the ones that I have shown you. Since they are different than intestinal helminths, their size simply is far, far smaller than all of the parasites we just took a look at. And so they have a lot more circumstances when it comes to how they actually survive. They typically need a water component. It's not easy for them to necessarily travel on their own just due to their size. So the oocysts, and we're going to be starting off with those instead of eggs, are going to be passed in the feces. And the feces has to be able to either infect the water or infect another organism that is close to the host. And once the definitive host is able to contract that parasite, that is when the actual reproduction and transmission of this parasite will occur. And it's important to note that the epithelial cells of the gastrointestinal tract are specifically affected by this protozoan because that shows you the size of it compared to residing in the small intestine. And it also goes hand in hand with all of the really nasty symptoms you get with a Cryptosporidium infection. Here you can see the life cycle. It is pretty simple. The bottom box is showing you the asexual as well as the sexual reproduction this organism can do. The life cycle is pictured at top, and you can see there's only one definitive host here. And that's what's so special about this parasite is that it really does need that water in order to be capable of infection. From my data, I collected 154 samples, and of those 154 samples, 30 of them had parasites in them, and that's roughly 20%. My main parasitic population are intestinal helminths, which are nematodes, trematodes, and cestodes. Here I have pictured my composition of my actual samples, and this is the parasitic composition. I have my main classifications there, and you can see there's a big blue chunk, and that are our tapeworms, and those are our cestodes. You can see a pretty big gray sliver, and that's actually arthropod, and we didn't talk about arthropods because they're a pseudoparasite for wolves. We typically don't find arthropods inside of organisms. They only like to live on the outside. So most likely these arthropods were parasitic to rabbits, and through the ingestion of the fur, that's how I was able to find the arthropod. Here you can actually see the diversity of my samples. These colors are representing each observation I made. So you can see at the bottom I have each sample that had a parasite in it. And the number on the left is showing how many observations I made from that sample. For example, the first one, you can see that it had a soft texture to it as well as a dark color. And for my observations, I was taking down the color as well as the texture and any elements that were included in that, whether it be bones, organs, anything I was finding. I actually found a rat tail at one point. But it's kind of important to see the observations made with the actual samples themselves, because you can see on this graph, there's a very big correlation with dark color and the amount of parasites found inside, as well as soft composition and hair. And it makes sense that the soft stool as well as the hairy stool would contain parasites, because wolves like to actually ingest grass and other substances that'll help naturally scrub out their intestines. So when they feel like they are sick with an intestinal helminth, they'll start eating grass in order to scrub their intestines clean of that parasite. And actually, in that cestode sample I found with the whole tapeworm, there was a ton of grass in that sample, and the worm was all wrapped up inside of the grass. And it is a very effective way for these animals to survive. So it's very important to understand that I only had stool samples to look at. So I was only able to observe the parasites that are transmissible through feces. I wasn't provided any other body fluid, and parasites are pretty much able to hijack whatever they would like in our body. So I was only able to actually analyze the fecal parasites. And the size of the parasite is also very important. For the protozoans and the smaller sized parasites, it's very difficult to actually see them and make the identification, as well as the fact that they are so fragile. It's also important to understand the actual dynamic between a parasite and a host. Since wolves are being newly introduced into our population at a high rate, some of the parasites we see in wolves now we may not see later. If wolves are gaining any benefit from this parasite, most likely this relationship will not occur. The parasite is there to reap the nutrients of the organism, and the host doesn't necessarily benefit much at all from that parasite. For my collection methods, I was collecting specimens after they were sitting out in nature for quite a while. So that occurred with a lot of pseudoparasites. I actually probably had to learn more pseudoparasites than actual parasites for identification. And the collection method is also very important because as these parasites are going through their life stages, when I preserve them, that halts the life stage that they're at. And if they are released in the feces as a larva, like a cestode, they have the capabilities of getting away from the feces itself. So sometimes I'm only left with the eggs, not necessarily the whole parasites themselves, just due to how nature works. And also, I was performing my research in a hunting shed. So I had some squirrels that became my best friends. Every single summer morning, I would go say hi to them. So my lab space was as sanitary as it could possibly be with my squirrel friends around. I also had a 10% formalin mixture that I made myself, as Mr. Robert Welch also helped me create this formalin mixture. So it wasn't bought from a lab. We did make sure that we were using the correct quantities. However, it is hard to say if the preservative was actually spot on. As far as my research goes, I did see full preservation of my samples. So I do believe that the formalin did work. I also have learned a lot of techniques with microscopy identification. The big part of microscopy is actually understanding the size of things, and this is especially important with parasites. The size of a parasite is going to be one of the biggest characteristics to identifying a parasite. Once we have the actual size of the parasite, we can then look at the contents within the egg to identify what it is. To make sure that I was thoroughly looking at my slides, I like to use a checkered technique on my slides, where I would do about a 4x4 kind of checkered idea on my slide, and that allowed me to look in each of those boxes specifically to make sure I was covering the full surface area of each slide I made. Excuse me one second. So like I was saying, pseudoparasites are a huge part of my research. I probably had to learn about more pseudoparasites than actual parasites themselves. I encountered many, many interesting organisms as well as just materials left in the feces. You can see pictured at top is a pollen grain, and they pretty much look exactly like that, almost all of them. I found quite a few pollen grains. At first, when I found them, I thought they were super fascinating, and then I learned it's just a pollen grain. You can also see on the bottom, I have pictured a plant hair. I found quite a few plant hairs. It kind of looks like a nematode. However, for nematode identification, it's really important to look at the digestive tract going through the worm. You should be able to follow a clear digestive tract from the mouth all the way to the end. And as you can see on the bottom, there isn't necessarily a clear digestive tract in that. So what does this really mean for wolves? Well, it's very important to study parasites because it tells us a lot about our environment. There's a lot of contamination aspects to what's going on in our environment, and just the ways that the parasites utilize our environment tells us a lot about what's actually occurring. With that, our environment then goes hand in hand with the health of our organisms, and we like to ensure that we do have a healthy wolf population. A healthy wolf population ensures that the parasite population will increase in a way that is beneficial to every organism within the environment itself. It's also important because, as you can tell from the life cycles, there are a lot of dynamics between certain organisms in their environment. And once we can actually look at the dynamics of those organisms, we can be able to tell a lot more of life cycles of each of those organisms involved, as well as what kind of steps we can take for protection for ourselves as well as our canine friends here. So through this, I have had a lot of personal growth as well as scientific growth. I am, like I said, a full-time student at UWSP. I also work part-time, and so it was really fun able to dedicate my summer to doing this research. And I was able to take on a lot of extra skills that I never thought I would acquire at the Wapaka Biological Field Station. This is a picture of me doing bird banding there with Robert Welch. I am terrified of birds, and this will probably be the first and last bird that I ever hold. I also grew quite a bit scientifically. I was able to really use all of my prior knowledge that I had gained and add on to that, and not only almost try to perfect my laboratory skills, but also gain so many more laboratory skills. And with that, I actually have never taken a parasitology course or anything related to parasites. Parasites are simply my own passion, and all of the parasitic knowledge I know is simply based off of my own research I have done. So what would a chapter 2 look like for my research? Well, I would really like to gain additional data. As you can see, I do have 154 samples, but it's always good to get more data, especially since 20% of those actually had parasites in them. Once I can actually gain the additional information, I can do further identification and classification of these parasites. It's important to make sure you're getting consistent data throughout the whole entire research process, simply due to the fact that once you have more to go off of, you can actually make statistical significances on the data that you're finding. And I would like to really determine if there's a statistical significance in the correlations that I found associated with the actual scat that I had and the parasites that I found. I would like to give a special thanks and acknowledgement to Robert Welch and Hanna Bukevich. This research could not have been done without them, and they have shown me so much guidance and help through this whole entire process.
Thank you, Lettie.
Thank you.
I'm going to announce the telephone number if anybody out there in listening world wants to ask a question of Lettie. That number is 715-942-9917. So here's the first question. Are the parasites you found, are they all harmful to the hosts?
So that's actually a very good question. Parasites have this very fine line of being able to use the nutrients of the host, but they don't necessarily want to kill the host. If they kill their host, they're not going to have a home to live in. So they want to be able to infect that host and use the nutrients without actually completely depriving the host, so they won't actually have a home to live in.
So are there any helpful parasites?
That is research that's actually being done. One of the research studies that I've actually been following for probably about 5 years now is actually looking into how roundworms are able to, and those are nematodes, are able to help people that can't necessarily live their life normally due to allergies. There's a specific white blood cell that deals with allergies and with roundworms, and if we're kind of able to trick that white blood cell into being preoccupied with the infection that's occurring, these people may be able to live their life allergy-free.
Interesting. So are there any specific diseases associated with the parasite infections, or is that the disease itself?
So that would be considered the disease itself. There's many side effects that come to having a parasite, and that is specific to the intermediate host. So when we're talking about an intermediate host like a wolf or a human being, it's not that we're the definitive host, so we're not necessarily the prime environment for that parasite to live in. We're just a step. We're just a little bus ride for them to their next destination. So they're not actually, can you repeat that again?
We were talking about whether they caused any other diseases.
Yes, sorry. So they, obviously, using your body, they are able to cause many side effects and symptoms, but overall, typically there aren't many diseases specifically associated with the parasite. The parasite would be the disease itself.
Okay, great. Have your results been published, or do you intend to submit them for publication anywhere?
I do intend to. I'm actually hoping to collaborate with Hannah Bukevich and work on a publication with her.
Great. Was your research funded?
It was funded. It was funded by the Deborah Ann Martin Memorial Fund Grant Program.
And where does that come from?
So that was actually given to me out of the Wapaka Biological Field Station through Robert Welch. It is a memorial fund that's in place for his wife that passed, and she wanted to ensure that female in STEM programs were able to actually perform the research that they would like to and advance their knowledge.
Wonderful. Maggie's got a question coming in from the phone. You can read it, Maggie.
This question kind of goes to the background of the research you did. How did you get interested in this Wisconsin wolf population and doing parasitical studies?
So that's actually a really good question. Around age 10, I watched an episode of Monsters Inside Me, and that really opened up my eyes to a whole world of life that I have never seen. I only experienced life as a human being, and to see the way that a creature, and it was actually an amoeboid parasite, so very, very small, single-celled, and the fact that something so small could take over such a large organism and use it for its benefit just absolutely fascinated me, and I've loved parasites since.
Well, maybe that goes a little bit toward this next question, too. Could you tell us about actually collecting those specimens? And I guess I would think about the yuck factor in collecting all that. And was that an issue, or is that just a scientific thing that you looked at it that way?
Yeah, so I would say in the beginning, it was definitely difficult getting used to. I do love parasites. I don't necessarily enjoy the process of gathering those parasites. And as far as the actual sample collection methods, it's a little bit hard for us to get into specifics with it just because it is a population that we're trying to protect. But as far as the actual collection itself, there was many times where I needed to step away from my table and kind of get a breather. Luckily, I was wearing a mask the whole time, which really helped. But a lot of them, especially the ones with parasites, have very distinct smells to them.
Well, here's one kind of off the top of my head, kind of along the same lines. Are any of these visible with the naked eye, or they all require a microscope to see?
Yeah, so there's quite a few parasites that are visible to the naked eye, and those parasites are going to be mainly the nematodes and cestodes that I talked about, and those are mainly the helminths are going to be the easiest to see with the naked eye. Compared to that, a lot of parasites you aren't going to be able to actually see.
Okay. Why don't we just rest for a minute or two and see if any other questions come in? So you mentioned that you haven't actually taken any classes on parasites. Are there any available that you can take before you graduate?
Yeah, so I plan on, and that's actually, I next semester plan on taking quite a few parasitology courses. That's one reason why I chose UWSP for my biology major, because I really am able to pick and choose the classes that I want to take. And next semester, I do plan on finally taking some parasitology courses.
Great. Well, thank you very much, Lettie. This was a fabulous presentation, and best of luck on your academic and research careers.
Perfect. Thank you so much.